How do anabolic and catabolic processes form the core of metabolic pathways? Published by sites.google.com. Catabolic pathways are characteristically energy-yielding, breakdown products, exogarnic, so this process releases energy. Anabolic pathways are characteristically energy-requiring, build products, endogarnic, which means the process requires energy. Catabolism involves the oxidative degradation of complex nutrient molecules. Anabolism is a synthetic process in which the varied and complex biomolecules are assembled from simpler precursors. Anabolism and catabolism are interrelated. Products from one provide substrates for the other. Many intermediates are shared between anabolism and catabolism. 2. Anabolism and catabolism are not mutually exclusive. Catabolic pathways converge to a few end products. Anabolic pathways diverge to synthesize many biomolecules. Some pathways are amphibolic, which serves both in catabolism and anabolism. 3. Comparing pathways. Anabolic and catabolic pathways involving the same product are not the same. Some steps may be common to both. Others must be different, to ensure that each pathway is spontaneous. This also allows regulation mechanisms to turn one pathway on and the other off. 4. The pathways of catabolism converge to a few end products. The 3 stages of catabolism. Stage 1. Proteins, polysaccharides, and lipids are broken down into their component building blocks. Stage 2. The building blocks are degraded into the common product, the acetyl groups of acetyl-CoA. Stage 3. Catabolism converges to 3 principal end products: water, carbon dioxide, and ammonia, in addition to the 2 end products of fermentation, lactate and ethanol. 5. Metabolic regulation requires different pathways for oppositely directed metabolic sequences. Parallel pathways of catabolism and anabolism must differ in at least one metabolic step in order that they can be regulated independently. Shown here are 2 possible arrangements of opposing catabolic and anabolic sequences between A and P. Parallel sequences proceed by independent routes, having different regulatory steps. Only one reaction has 2 different regulatory enzymes of specific step. 6. ATP serves in a cellular energy cycle. ATP is the energy currency of cells. Phototrophs transform light energy into the chemical energy of ATP. In heterotrophs, catabolism produces ATP, which drives activities of cells. ATP cycle carries energy from photosynthesis or catabolism to the energy-requiring processes of cells. 7. The ATP cycle in cells. 8. The substrates of catabolism contain relatively reduced forms of carbon. Comparison of the state of reduction of carbon atoms in biomolecules. Chains of CH2 groups are the most practical form of reduced carbon in the biosphere. Carbon dioxide is the final product of catabolism and the most oxidized form of carbon in the biosphere. Compare an individual C, with its associated H, O. 9. NAD+ collects electrons released in catabolism. The substrates of catabolism, proteins, carbohydrates, and lipids, are good sources of chemical energy because their carbon is reduced. The oxidative reactions of catabolism release reducing equivalents from these substrates, often in the form of hydride ions. These hydrides are transferred to NAD molecules, reducing them to NADH. NADH in turn passes these reducing equivalents to other acceptors. The ultimate oxidizing agent, O2, is the final acceptor of electrons, becoming reduced to H2O. Hydrogen and electrons released in the course of oxidative catabolism are transferred as hydride ions to the pyridine nucleotide, NAD+, to form NADH plus H+ in dehydrogenase reactions. 10. NADPH provides the reducing power for anabolic processes. Whereas catabolism is oxidative, anabolism is reductive. Biosynthesis is typically reductive and requires reducing equivalents, from NADPH. NADPH can be viewed as the carrier of electrons from catabolic reactions to anabolic reactions. In photosynthesis, light energy is used to pull electrons from water and transfer them to NADP+, in which O2 is a byproduct of this process. Transfer of reducing equivalents from catabolism to anabolism via the NADPH cycle. 11. A summary of vitamins and coenzymes discussed elsewhere in the text. Question-related statements. Anabolism is biosynthesis. As such, it is endergonic. Anabolism and catabolism are not mutually exclusive. The pathways of catabolism converge to a few end products, such as lactate, ethanol, CO2, H2O, and NH4. Amphibolic intermediates play dual roles, serving roles in both catabolic and anabolic pathways. During catabolism, energy-rich electrons are transferred to the coenzymes NAD and NADP. ATP serves in a cellular energy cycle, being produced in catabolism, or photosynthesis, and consumed in biosynthesis, or osmotic work, or motility. The energy released in NADH oxidation is coupled with the formation of ATP. The coupled process is called oxidative phosphorylation. Transformation of the energy released in an oxidation-reduction reaction into the chemical energy of a phosphoric anhydride bond is a form of energy transduction. Water-soluble vitamins are precursors for coenzyme synthesis.